Slide 1, Biochemical Assessment of Nutritional Status, Part 2. Hello everyone, and welcome back. In this lecture, we'll continue our exploration of biochemical assessments of nutritional status, focusing on specific micronutrients. I'm Dr. Nadeem, and I'm excited to guide you through this important topic. Today, we'll be diving into the biochemical assessment of three key nutrients: iron, calcium, and zinc. We'll discuss how we can evaluate the status of each of these nutrients in the body using various laboratory tests. This will build upon the general principles of nutritional assessment we covered previously. By the end of this lecture, you should be able to confidently discuss the assessment of iron, calcium, and zinc status. This includes understanding the relevant biochemical markers, their interpretation, and their clinical significance in identifying deficiencies or excesses. As a reminder, nutritional assessment involves a multifaceted approach. We use anthropometric, biochemical, clinical, and dietary methods to get a complete picture of a person's nutritional health. Today, we're focusing specifically on the biochemical methods which provide objective measures of nutrient status. Iron is an essential trace element vital for oxygen transport, enzymatic activity, and DNA synthesis. It's a key component of hemoglobin in red blood cells and myoglobin in muscles. The majority of iron in the body is found in hemoglobin, with smaller amounts stored as ferritin and hemosiderin and used in enzymes. Iron absorption primarily occurs in the duodenum and upper jejunum. Heme-iron, found in animal products, is absorbed more efficiently than non-heme iron from plant-based foods. The absorption process involves conversion of iron and transport across intestinal cells. Once inside the enterocytes, iron is transported to plasma via membrane protein known as ferroportin 1. Iron, Siku, is immediately to iron, the third, in the blood plasma by a protein known as hephaestin or ceruloplasmin. Iron, the third, binds to transferrin and transported to the all-body cells for their requirements. If the body doesn't need iron immediately, it's stored as mucosal ferritin within the intestinal cells. This stored iron is then lost when these cells are shed, representing a mechanism for regulating iron levels. Hepcidin, a hormone produced by the liver, plays a central role in regulating iron levels. It blocks ferroportin 1, preventing iron from being released from enterocytes into the bloodstream, thus controlling iron absorption. Iron exerts its effects primarily through the molecules it's incorporated into. Hemoglobin and myoglobin are essential for oxygen and carbon dioxide transport, while iron-containing enzymes are involved in various metabolic processes. Iron deficiency is the most prevalent single nutrient deficiency worldwide and the leading cause of anemia. Preschool children and women of childbearing age are particularly vulnerable. It can result from poor intake or absorption, blood loss, or increased iron requirements during growth or pregnancy. Iron depletion progresses through three stages. The first involves depleted iron stores without any adverse effects, followed by early functional iron deficiency without anemia, and finally, iron deficiency anemia. Each stage is characterized by specific changes in biochemical markers. Serum ferritin is a primary storage form of iron and a sensitive indicator of early iron deficiency. As iron stores decline, serum ferritin levels decrease. However, it's important to note that ferritin is also an acute phase protein, meaning its levels can be elevated during inflammation, potentially masking iron deficiency. Serum ferritin is a valuable marker for assessing iron status. It reflects total body iron stores, serves as an early indicator of iron deficiency, and can also help identify iron overload. However, its interpretation must consider the presence of inflammation or chronic disease. Normal serum ferritin ranges vary by population group. Low ferritin levels typically indicate iron deficiency, while high levels may suggest iron overload or inflammation. Understanding these ranges is crucial for accurate interpretation of test results. Transferrin is a glycoprotein that binds and transports iron in the bloodstream. It plays a critical role in iron homeostasis by regulating iron uptake, storage, and utilization. Measuring transferrin levels is essential in iron assessment to diagnose iron deficiency, anemia, and iron overload disorders. Transferrin acts as an iron transporter, delivering iron to cells, particularly in the bone marrow for red blood cell production. Transferrin levels rise in iron deficiency and decrease in iron overload or inflammation. It's also used to differentiate between different types of anemia. TIBC reflects the total amount of iron that transferrin can bind, while transferrin saturation indicates the percentage of transferrin that is actually bound to iron. These measures provide additional insights into iron status. In iron deficiency anemia, serum iron levels decrease and TIBC increases, resulting in a decreased transferrin saturation. Transferrin saturation is calculated as the ratio of serum iron to TIBC, expressed as a percentage. Low transferrin saturation suggests iron deficiency anemia, while high saturation indicates iron overload. Soluble transferrin receptor, STFR, is a shortened form of the membrane-bound TFR that is cleaved from TFR and released into the serum. The concentration of STFR is proportional to the concentration of TFR on the plasma membranes of cells, especially the erythrocyte-producing cells of bone. The STFR concentration begins to increase in early iron deficiency with the onset of iron-deficient erythropoiesis, red blood cell production, before anemia develops and continues to increase as iron-deficient erythropoiesis worsens. Serum STFR concentration is not affected by concurrent inflammation or infection. Erythrocyte protoporphyrin is a precursor of heme that accumulates in red blood cells when iron is deficient. Elevated levels indicate that the body is unable to produce sufficient heme due to lack of iron. While hemoglobin is commonly used to screen for iron deficiency anemia, it's not a reliable indicator of early iron deficiency. Hemoglobin levels tend to become abnormal only in the later stages of iron depletion. Mean corpuscular volume, MCV, is the average volume of red blood cells. Factors decreasing MCV, resulting in microcytosis, are chronic iron deficiency, thalassemia, anemia of chronic diseases, and lead poisoning. Accurately assessing iron status can be challenging as no single test is perfect. Therefore, it's recommended to use a combination of biochemical indicators to get a comprehensive picture. Now let's shift our focus to calcium and zinc. We'll explore their roles in the body and how we can assess their status biochemically. Calcium is the most abundant mineral in the human body, with the majority found in bones and teeth. It's essential for bone health, nerve transmission, muscle contraction, and various other functions. Milk is an excellent source of calcium, and adults need around 800 milligrams per day. Normal calcium level in blood is 911 mg/dl. Free form is the biologically active form, bound with protein albumins and to some extent with globulins. Calcium plays a crucial role in the development of bone and teeth. Bones act as a calcium reservoir, with osteoblasts promoting bone deposition and osteoclasts facilitating demineralization. Calcium helps in the transmission of nerve impulse by release of neurotransmitters into synaptic clefts. Calcium plays an essential role in the muscle contraction. Ca binds with troponin, which is attached with tropomyosin and helps in the activity of actin and myosin in the muscle contraction.